Did ancient DNA just confirm what the Bible has been saying about the Jewish people for 3,000 years? A 2024 study pulled genomes spanning millennia from across the ancient Levant and uncovered something extraordinary. Every major Jewish population on Earth, from Ethiopia to Eastern Europe, carries a shared ancestral signature traced to one source region. But what that signature reveals about exile, survival, and identity is where this story truly begins. In 2024, a landmark study published in the journal Nature dramatically expanded our understanding of historical genetics. A global team of researchers analyzed more than 700 ancient genomes, spanning a chronological range of nearly 4,000 years. This massive dataset allowed scientists to reconstruct the population history of the Near East with unprecedented precision, bypassing speculation to look directly at the physical remains of those who lived and died in the region. The genetic data pointed to one primary source: the southern Levant, a geographic area overlapping modern Israel and Jordan. When researchers compared these ancient profiles to modern populations, they found a highly consistent pattern of continuity. Across geographically separated communities, modern Jewish groups, including Ashkenazi, Sephardi, and Mizrahi populations, showed between 50 and 80% of their ancestry linked directly to Bronze Age and Iron Age Levantine peoples. This is a structured, persistent signature, not a loose correlation. This is population history reconstructed through genomic science, independent of theological claims. Yet, the genetic coordinates map directly onto the geographic center of historical Jewish memory. This is not Sunday school. This is ancient genomics reading the same story the biblical scribes recorded in the ancient Levant. By measuring the genetic drift and ancestral inheritance preserved across millennia, the study provides a biological archive of migration, exile, and survival. To read these ancient genomes, scientists target the densest structures of the skeletal remains, the inner chamber of the petrous bone in the skull, and the protective cementum of tooth roots. Here, microscopic fragments of DNA can survive for thousands of years. Before sequencing, researchers apply a strict credibility gate, screening for contamination by analyzing chemical decay patterns at the ends of the DNA strands, a signature that proves the genetic material is ancient rather than modern. Once authenticated, the raw data is mapped using advanced statistical tools. Software models estimate admixture proportions, calculating ancestral contributions from distinct source populations. Beyond basic percentages, researchers analyze haplotypes, large, unbroken blocks of inherited DNA that reveal shared descent across geographically separated communities. By measuring how these blocks break down over generations, molecular clock reasoning and founder event modeling date precisely when ancestral groups split or collapsed. Scientists do not rely on a single statistic. They cross-reference these findings across multiple independent genomic methods, ensuring the historical map they reconstruct is mathematically sound. For decades, a politicized theory attempted to sever the genetic link between modern European Jews and the ancient Middle East. First popularized for modern audiences by writer Arthur Koestler in his 1976 book, The 13th Tribe. This hypothesis argued that Ashkenazi Jews descended from the Khazars, a Turkic-speaking Caucasian population that converted to Judaism in the 8th century, instead of ancient Israelites. This claim quickly became a weapon in geopolitical debates, used specifically to delegitimize Jewish historical ties to the land of Israel. The 2024 genomic data dismantles this narrative. When researchers mapped the Ashkenazi genetic profile, they found a genome that is roughly half Middle Eastern and half Southern European, with the European portion centering around Italy. The genetic signature from the Caucasus is less than 5%. In population genetics, a trace that small does not indicate mass descent from a medieval steppe empire. Instead, it fits the pattern of ordinary background admixture expected in a vast Eurasian trading world where merchants, travelers, and families moved along established trade routes. In 2013, geneticist Duran Bihar published a genome-wide analysis in the journal Human Biology, showing no ancestral link to the Caucasus. The 2024 study confirms this finding on an unprecedented scale. By comparing modern genomes with over 700 ancient Near Eastern samples, the statistical models show no distinct Caucasus source. The biological core of the Ashkenazi population remains rooted in the southern Levant, burying the Khazar theory under the weight of empirical data. But this ancient Middle Eastern core also preserves the physical signature of a catastrophic demographic collapse. Between 600 CE and 800 CE, the ancestral Ashkenazi population underwent a severe bottleneck. The breeding pool shrank to an estimated 350 to 500 founders. As this small population slowly rebounded through centuries of endogamy, marrying strictly within the faith, genetic drift amplified rare mutations, creating a dramatic founder effect. Today, this historical crash is written directly into Ashkenazi medical genetics. Specific mutations became unusually common, resulting in high rates of recessive disorders like Tay-Sachs and Gaucher disease, as well as the BRCA1 and BRCA2 mutations linked to hereditary breast and ovarian cancer. Modern screening programs and commercial databases like 23andMe now flag individuals of Ashkenazi descent for more than 40 hereditary conditions based on this shared genetic heritage. By mapping long, identical by descent blocks of DNA, researchers can trace these medical risks directly back to that medieval crisis. Yet, this collapse did not create a new ancestry. It merely concentrated what was already there. Beneath these common disease variants lies the same unbroken, historic Levantine genome, carried through the medieval bottleneck and preserved across generations. The genetic narrative takes a distinct turn in the Horn of Africa. For centuries, the origins of the Beta Israel, the Jewish community of Ethiopia, remained isolated from the European diaspora. Traditional accounts often treated their heritage as a purely cultural adoption. However, ancient genomics reveals a different reality. Beta Israel genomes carry approximately 30% Levantine-related ancestry, a genetic signature far more pronounced than in neighboring, non-Jewish Ethiopian populations. This genetic link indicates physical migration and historical contact. Ancient trade corridors across the Red Sea and the Gulf of Aden once linked the East African highlands directly to the biblical world, a network mirrored in the commerce of 1 Kings chapter 10. Yet, this ancestral signature marks the boundary between genetics and faith. Population genetics can trace ancient migrations, but it does not define religious belonging. For the Beta Israel, historical identity remains anchored in centuries of devotion, separate from the percentages written in their DNA. In Iraq, the genetic profile of the Jewish community reveals an unbroken link to ancient Mesopotamia, matching the historical timeline of the Babylonian exile that began under King Nebuchadnezzar II in 597 BCE. Genomic comparisons show these populations retain a striking similarity to Iron Age Israelites, making them some of the closest living genetic relatives to the people of the ancient biblical kingdom. Further west, Sephardic Jewish communities in Turkey, Greece, Morocco, and the Netherlands display almost identical genetic structures, despite being scattered across the globe after the Alhambra decree expulsion of 1492. Mizrahi populations show an even higher concentration, preserving over 80% of their ancestry from Bronze Age Levantine peoples. This persistent genetic signal acts like a molecular clock, tracking migrations and expulsions across generations. This high level of genetic preservation is driven by centuries of endogamy, marrying within the community, which prevented genetic drift from altering their Middle Eastern baseline. Geneticist David Reich describes Jewish populations as exceptional in their balance between isolation and migration. Just as the genealogies of Genesis chapter 10 map out a world of shared descent dispersing from a common origin, the ancient DNA maps a matching biological path, showing how a scattered people carried their physical homeland with them into every corner of the earth. Science has transformed a theological mystery into a biological reality. This is not Sunday school. This is ancient genomics reading the same story the scribes wrote down in the ancient Levant. For a diaspora once defined by exile, the dust has finally spoken. Share your thoughts below.